Hello, my name is Andrii Domanskyi from joining you from Orion Corporation in Turku, Finland. And today we will be talking about pain and about influence of inflammatory system and inflammation in chronic pain. But at the beginning, a few words about myself. I've been working in academia for over like 20 years, and since 5 years ago, I switched from working at the University of Helsinki, Finland, to working Orion Corporation, which is a pharmaceutical company in Finland, and started with continuing with my actual work in neurodegeneration. I've been working a lot with Parkinson disease models and have quite extensive academic experience with these Parkinson disease models. And since 3 years ago, I switched my focus to working with pain. And hence, this is the talk which I'm giving you today. So we are interested in developing compounds and drug treatments for chronic, neuropathic, and musculoskeletal pain. And then I'm as a vivo scientist, mostly involved in vivo rodent models of pain, especially osteoarthritis pain, as well as ex vivo and in vitro models of pain and activation of sensory neurons, for which we will be actually talking through this presentation. I've been asked to present you learning objective. So during this presentation or after this meeting, hopefully we will be able to define some or have some idea and define pain neuronal circuitry and the role of non-neuronal cells in pain cessation, which we will specifically discuss immune cells, especially macrophages in chronic pain. We will, of course, touch upon preclinical models. We will be talking about specifically rodent preclinical models of pain. It's not solely, pain is not only the model in rodents, but for the sake of the time, we will focus on rodents today. And then we have some examples of clinical translatability, problems, issues with clinical translatability of results of preclinical studies towards clinical applications and drug development. So, pain. What is pain? If we take a textbook or actual updated textbook definition of pain, it's wrong that this is unpleasant, sensory, and importantly, also emotional experience associated with or resembling that associated with actual or potential tissue damage. So it can be responding to actual tissue damage, or it can be responding to potential tissue damage. And there are different types of pains, and there are different ways how pain is processed in our body. And what happens actually when we encounter some noxious stimulus? So, well, for example, in this particular case, on your left, you see the scheme of pain processing pathways in the body and the brain. So after sensory stimulation, in this case, nail hitting your hand, there are receptors which are activated on your skin, and then in the tissue there is also tissue damage occurring in these receptors, and the signal from sensory neurons is transmitted through their axons through cell bodies in dorsal root ganglion to dorsal horn of spinal cord, and then through ascending pathway to the brain, through medulla and then midbrain towards thalamic structures. These thalamic projections, they rely to then higher brain areas like cortical centers, where emotional processing of pain is occurring. And then on your right, you see the scheme of pain processing network in the brain. Those are thalamic and other structures, brain regions involved in different aspects of pain processing. And then, importantly, there is also, so in addition to this ascending pathway, pain is modulated through this loop, so this descending pathway, which is then projecting down through the periaqueductal gray area in the brain, and then down to rostroventral medulla region, back to spinal cord, and the loop is enclosed in the dorsal horn. So in this way, pain is really combining sensory perception with modulation, so the sensory perception which is modulated by higher brain areas, including emotional component. On the molecular, on the cellular level. So how neurons are actually sensing this pain. So on the cellular level, neurons, or sensory neurons, they express different receptors. There has been a lot of studies previously, or like, and still continuing to discover receptors for different stimuli, such as cold, heat, acid, or mechanosensitive receptors. There are Nobel Prizes awarded for the discovery of these receptors, and the work is actually continuous. But most of the receptors are now discovered, and these receptors are, by binding to their, or being activated, these are ion channels, so they increase conductivity of different ions, mostly calcium, which then changes membrane potential of the sensory neurons, leading to generation of axon potential, which is then transmitted to the pathways we were describing on the previous slide. When you have tissue damage, so a cut of your finger, immune cells are activated. There are several types of immune cells. For the simply for illustration purposes, we are here having macrophages and mast cells. So they release immune modulators, immune mediators, such as cytokines, prostaglandins, which are then interact with corresponding receptors on sensory neurons, and again by either directly modulate those receptors. So this receptors, like for example, the TRPV1 channel. So when you see a scheme in the middle of the slide, or by activating intracellular cascades. Anyway, leading to again changes in neuronal membrane potential and generation of axon potential propagating signal to the spinal cord. Interestingly, for example, here you can also see cyclooxygenase 2, this COX-2 enzyme, which is producing prostaglandins, and this is targeted by non-steroid anti-inflammatory drugs like ibuprofen or celecoxib. So very common painkillers for inflammatory pain. And then in terms of neuropathic pain, which what you see the scheme on your right, this occurs when there is a neurodegeneration, when the neurons are cut or the axons are degenerating. So there is also activation of immune cells in response to degeneration of neurons, which then act on surviving neuronal fibers, activating their receptors and pain sensation through those neuronal fibers, leading to this so-called neuropathic pain. How we know that? how do we actually study this pathway? I can present you a lot of data and describe a lot of things, how we have like how they are discovered, but I just wanted also to share with you some ideas of how actually these processes have been studied. So, for example, in our unit, we are using both in vitro and in vivo models. And it's not only us, there is a lot of other scientists who actually approach this from different options. There is a possibility to use primary cultures of rodent neurons. There is like we can cut dorsal root ganglia. We can also culture spinal cord neurons. These neurons can be cultured either alone or in combination with other cell types like macrophages, oligodendrocytes, mast cells, uh, microglial cells, and then these cultures can be subjected to treatment of different compounds, cytokines, viral vectors for genetic tools, for analysis of their effect on neuronal excitability, which can be studied using electrophysiological methods, multiple electrode array, immunological, like immunostaining, so we can study protein changes in protein expression, or all kinds of omics assays for analyzing protein levels and changes in response to different and molecular pathways which are responding to different treatment options. The more recent advance is use of human-induced pluripotent stem cells and differentiated to sensory neurons. It's not that easy to differentiate stem cells to sensory neurons, and corresponding protocols have been developed during the maybe last 5 years or so, but currently they're available, and we can also study human. The advantage, of course, of this method is that you study human sensory neurons in contrast to rodent sensory neurons, which are used by previous time. We can also do primary human neurons, which unfortunately cannot be taken from like people, from like living humans, and have to be taken post-mortem, and therefore they are not much available. And there are just some examples of neurons which are in culture, sensory neurons in culture from our laboratory. In vivo pain models can be, and now again we are talking about rodents, so there we can model different aspects of pain, being inflammatory pain, so we can inject some compounds which are inducing inflammation response. One of the most popular models is injection of this complete Freund's adjuvant. This is adjuvant, which is used also in vaccines, which by local injections would induce immune reaction and causing pain. You can also induce inflammation in the joint, and in this way we will be modeling some more like osteoarthritis, inflammation during osteoarthritis pain. Neuropathic pain, as we discussed, molecular mechanism. So this is like neuropathic can be modeled by physical damage of inflicting physical damage of neurons, like we are, for example, using this chronic constriction injury model. There is also, you can ligate nerves and do several ways of surgery, several surgical models to actually induce this nerve injury, and this is schematically represented on your right slide. Musculoskeletal osteoarthritis. This is my kind of favorite topic, which I'm working mostly with. You can inject it either use again surgical models to destabilize knees and model osteoarthritis progression and degeneration in the joints by using surgery, or by injecting different compounds such as monosodium yogurt acetate to the knee. And this is this inset on your right, where you see this MIA injection to knee joint of a rat. There is also cancer pain, and it's very well known that chemotherapy can induce neuropathic pain. And there are several cancer drugs like oxaliplatin, and this is very well known side effects of the cancer drug. And this also can be modeled in rodents to study cancer pain, and therefore also test compounds which will be blocking these types of pain in animals. One of the obvious problem with pain assessment is that we cannot really ask rodent about how it feels, how is it painful or not. And another problem is that actually rodents are evolved in hiding their pain, so they are not really showing that they are being in pain, even though they may be. So there are several ways how we can try to assess this pain in rodents. We can stimulate their legs, their paws with mechanical filament, so with filaments, and measure with different strengths, and measure the time of the strength when the animal uses responding from like removing their pain, their paw from filament stimulation. We can stimulate with heat, we can stimulate with pressure, and this is on this panel, like bottom panel of your left. You can press the knee, and then which evokes like vocalization response, or again withdrawal response of the legs, of the paw. There are also cold evoked stimuli or temperature preference. We can measure how animal behave if we induce either like ice, so like apply cold to the paw and again measure paw withdrawal reflex, or another possibility is quite frequently and relatively straightforward ways to use this acetone test, which is so acetone is due to fast evaporation. It also creates this cooling effect, and the animal would respond by licking its paw, which can be measured by the time amount of which is leaking. And we assume that these responses are well changing in response to pain. So when the animal is pain, they apparently, and like this is validated with painkillers, this is so apparently the timing of this response is very different whether animal is in pain or this is not like a control animal. So for example, it can withstand higher pressure or stronger heat compared to normal. There are non-evoked pain, and this is most interesting part, which is unfortunately also most difficult to implement. So we can also study non-evoked responses in rodents, and like changes in their grimace, so the changes in facial expression. There is also some in rodents, especially in mice, there is this burrowing measures. So they, when the animal is in pain, they try to dig more actively, and this can be even quantitatively assessed. Weight balance. So usually, if we inflict model like osteoarthritis model in one of the hind paws, which is normally done, the animal can go put its balance to more healthy paw. And gait changes, and actually this particular example has been already described some years ago, and at that time there was already some speculation that you can use videotaping of behavior and some kind of computational analysis to analyze behavior of animals in their home cage and to see if there would be some kind of changes in response to when the animal in pain or not. And the latest development in this direction is a device. So this is called this black box device. Is one of the several ones. So this one, like has been used in several applications. It's been published several years ago, and it's actually quite interesting way to analyze unbiasedly and objectively by using artificial intelligence based or machine learning based image analysis. So the animal is videotaped from below, behavior is videotaped, and also the pressure of paw against the glass can be measured by using clever algorithms. I encourage you to read their publication, which is like link you can see on your right lower corner. And what is interesting that when you inflict some nerve injury, in this case, you can see a difference between usage of the paws from like hind ipsilateral, so which was injured versus non-injured paw. So you see this red trace is now reduced low. So now you should look at your left lower graph, and then when you have the spared nerve injury. And in this case, just by videotaping animal with very relatively straightforward procedure, you can start observing those difference in pain behavior. And this is very useful and quite robust and reproducible way to automate pain behavioral assays in rodents, which we are currently evaluating. Brain imaging is another way. So a PhD, is a very talented PhD student in our lab. He has implemented it in collaboration with our colleagues from university, where we studied brain response in a pain model. In this case, this is osteoarthritis pain animal rats, which have progressive osteoarthritis in their one of their hind legs, hind paws, and by stimulating this hind paw, we see a signature of a pain in the brain of the animal, and then we can study how pharmacological compounds would affect this pain representation in the brain. We can see whether it's specifically whether ascending pathway affected or descending pathway affected, and then also whether we can even try to understand this emotional processing or emotional component of the pain, which I was talking at the beginning. So brain imaging is a very useful tool. Unfortunately, it's quite expensive or like not very easy to use with, and it requires a lot of expensive imaging time. But the results which you can get from them is very interesting, and this is also have translational value towards human subjects because there are also like similar changes in brain regions are observed in human patients who are having chronic pain. Well, back to the scientific. So these tools are used to understand interaction between pain of between neuron sensory system or like modulation of pain and other systems in the human body. Several years ago, I was really excited when I saw publication of this review about comparing immune and sensory system. And it's very strikingly similar between those. They are very strikingly similar between these two systems. Why? While both of them, both immune system and sensory neuron system, they are designed, they are, well, not designed, they evolved to coordinate host defense, and they share similar, if not architecture, but similar logic. So they are sensing damage, they have, they can integrate and process damage signals, and they have memory. Immune system also have immune memory, so they implement it in different ways, but the goals of these both two systems are very similar, and actually there is a crosstalk between immune cells and sensory neurons, and this has been already for quite known for quite some time, using co-culture systems, using in vivo studies. In the middle, you see this in the middle diagram, you see how sensory neuron responds to injury, as we were talking before about this injury-induced pain, but also inflammation-induced pain. And there are different compounds, different cytokines which are released by immune cells, which can affect neurons to modulate their excitability and which would lead to pain sensation. On the other side, neurons, sensory neurons, they also release several components, mediators, like CGRP or bradykinin, which can then, substance P, which can then modulate immune cells and create, if you wish, this some kind of vicious cycle of neuroimmune interaction, leading to induced pain sensation. Such neuroimmune interaction lead to sensitization of sensory neurons, and well, sensitization in this case, so meaning that the threshold of activation of these neurons is reduced, so that stimulus, for example, stimuli which were not sensed as a painful for normal in normal conditions would start to be painful if the neurons have to be sensitized. There are several molecular mechanisms described for this neuronal sensitization. So again, cytokines can induce intercellular cascades, leading ultimately to modulation of a level of particular receptors on neuronal membrane, phosphorylation of these receptors, or activation of their activity. Again, reducing threshold of activation and inducing pain sensation. As a last couple of minutes, I just wanted to tell you almost success story. So can we target immune cells for neuroimmune interaction for therapy? And there is like several years ago, several companies designed antibodies which were blocking a neural growth factor. And neural growth factor is a factor which is involved both in pain and in neuronal sensitization, so the pathways I described you, and especially in osteoarthritis pain, it has been studied. This neuronal growth factor, when blocked, causes reduction of pain in preclinical models, and with antibody, which would be like blocking this factor and reducing its interaction with cognate receptor on neurons and also on immune cells, would lead to reduction of pain in preclinical models, which is those tests of pain tests, results of those pain tests are shown on your right side. Unfortunately, it turned out that in clinical trials, well, while there was a very profound analgesic effect, there was also observed in some small amount of patients, there was observed actual progression of osteoarthritis, or so-called rapidly progressive osteoarthritis. Mechanism of that is not fully not clear, not fully clear, but nevertheless, this unfortunately this anti-NGF antibody is not in clinical. It did not reach clinic. The clinical trials have to be stopped because of the safety concern. Again, demonstrating that unfortunately, not all aspects of not all safety aspects can be modeled using animal models, and eventual truth will be only discovered after clinical trials in humans. Last few words about macrophages. So then, can we target some other cellular components? And for example, it's known that anti-inflammatory macrophages can promote pain relief. And there, as again, here you can nicely see the scheme where they represent multiple compounds, multiple mediators which are released by these activated macrophages, which are interacting with neuronal receptors and promoting neuronal sensitization. Whereas if anti-inflammatory macrophages, so-called M2 macrophages, they are known to release compounds which are actually reducing inflammatory response, reducing release of pro-inflammatory cytokines, and by this way may reduce pain sensation. And this is very interesting area of studies. There are several preclinical or multiple preclinical studies demonstrating that actually this pro-resolving, also this anti-inflammatory or pro-resolving inflammation macrophages, they do have analgesic effect in preclinical models. Very interesting subject to study. So probably can it be a role for next compound of next successful drugs treating chronic pain? Can they target macrophages? Well, the time will tell. So in summary of today talk, I just, if you want to remember something from my talk, I want you to remember that pain sensation involves both sensory and emotional components, and then activation of sensory neurons can be studied both in vitro and in vivo, and there are ways to assess pain behaviors in rodents with advance of machine learning and artificial intelligence algorithms. It will become more and more easy to understand pain behaviors in rodent models, and hopefully also with brain imaging tools to develop better models and better readouts from these models, which will hopefully improve clinical translation of preclinical results. And immune system is affecting sensory neurons. Immune cells are important for pain mediation, and targeting of the immune system will not only affect inflammatory pain, as we know, for example, with non-steroid anti-inflammatory drugs, but also will be useful for more chronic pain. With that, I would like to thank you very much for your attention, and thank you very much for inviting me to give this talk.